And today we're going to be looking at the human reproductive system, which most of you have hopefully been doing, usually in the first term roundabout. Otherwise, this might be a very good lesson for you to catch up. And we're going to jump straight into it with the first slide, which tells us a bit about puberty. Because all of this really starts with puberty, and it's quite a hormonal story. Puberty makes little pudgy boys into handsome men, and it's all about the hormones that are involved. So this is a kind of an overview slide that we're going to be looking at. So you can actually refer back to this at a later stage, and a lot of the hormones that we're going to be mentioning now, we will mention as we go through the different structures in the reproductive systems. So a lot of terminology coming your way, but don't worry, we will recap as we go along. Okay, so puberty starts with the hypothalamus that releases a hormone telling your pituitary gland to switch on the action. And this gland now releases two hormones, the first one called FSH, which stands for follicle stimulating hormone, referring to its function in the female reproductive system, namely the stimulating of new follicles. It also causes the release of LH, or luteinizing hormone, again referring to the function that it has in the female reproductive system in the production of a corpus luteum. In males, FSH will then do something very similar to what it does in females, and that is stimulate the testes to produce sperm. And LH in men also are acting on the testes, causing it to release testosterone, the male reproductive hormone. And testosterone during puberty now leads to the development of secondary sexual characteristics. In other words, characteristics that you were not born with and that develop secondary after puberty. The beard and the lowered voice, etc., that makes you appear to be a man. FSH in females leads to the development of the ovum in the ovaries, which in turn then releases one of the female hormones, the important one, estrogen, which also acts in on the whole body cells to cause the development of secondary sexual characteristics, the things that make you look and appear to be a female, the development of breasts, the start of the menstrual cycle, the distribution of fat, etc. And then LH in females does two things. So high levels of this during the menstrual cycle will trigger ovulation, which is the release of an egg cell or an ovum from the ovaries, and the development of the corpus luteum out of the remnants of the follicle from which this ovum was released. That corpus luteum then in turn releases another important female hormone called progesterone, and all of these hormones, progesterone, testosterone, as well as estrogen, have negative feedback effects on the glands, the hypothalamus and the pituitary, because we can't just have these hormone levels going up and up. Something needs to control them. So this is a very classic example of negative feedback. And now we're going to jump into the male reproductive system, and Yolandi is going to take us away there.
Right, so for the section on the male reproductive system, if we jump into the first structure, we've got the scrotum, which is represented here in yellow on our diagram. Now, pretty much for the structure, its main role is to actually regulate the production, the temperature for healthy sperm production. So usually what we find is that the sperm's production needs to be at 2 to 3 degrees Celsius below body temperature, and that will ensure that the sperm cells that are formed will obviously be fully formed, functional, able to swim sperm cells. Now, it is important that when you stipulate the function of the sperm cell, that you state that it is for healthy sperm production, because even if the temperature was slightly higher, closer to body temperature, there will still be sperm production that takes place, but it might not be a healthy sperm that is formed. So we do have to emphasize in a testo exam that it's for healthy sperm production. Right, if we look at our second structure on our diagram, we have the testes represented here in red, and this guy is obviously the site where the sperm cells are produced. And in addition to that, it also produces the hormone testosterone that Marichen was talking about, which is released during puberty. Now, let's say, for instance, that they give you a scenario where the question states something like, what will happen to a person that wears tight-fitting underwear? You would have to relate to, obviously, how, if you have tight-fitting underwear, the scrotum would not be able to perform its job. Testicles will be too close to the body, which will obviously elevate the temperature, and it will be too warm for the sperm to fully form and produce healthy sperm cells. So that's a typical question that we've seen in past exam papers. However, you could also approach it from a pressure perspective, and a lack of blood circulation perhaps going towards the testicles, and that will also negatively affect sperm production. Right, so let's have a look at our next part. If we make a cross section through the testicle, as we've seen here, we now have a look into one of the seminiferous tubules. As we can see in the diagram, the testes is made up of many coiled tubes. Now, if we look into one of the seminiferous tubules, we will see the site where the sperm cells are produced. Now, usually what will happen is that during puberty, testosterone will be released, and that's usually the trigger point of where the sperm cells will start to produce. Now, the wall of our seminiferous tubules, where we find our germinal epithelial cells, and they are referred to as diploid cells. And then through the process of meiosis, we see the development of sperm towards the center of the tube, going through the several stages of meiosis. So we start off with having a diploid cell called the spermatogonium, and as it moves from obviously through the steps of meiosis, as we'll see in the next slide, it then develops into primary spermatocyte, then it's referred to as a secondary spermatocyte, eventually it forms a spermatid, and then right at the end we have our fully formed spermatozoa, or commonly known as a sperm cell. Right, so the question is, how are sperm cells actually made? Now, I've already made reference to the fact that sperm is produced through the process of meiosis, which is also a chapter that you would have already completed by this point. But if we are being very specific here, we would say that the formation of sperm cells through the process of meiosis is known as spermatogenesis. And when we are talking about the formation of an ovum or oogonium, a fully formed egg cell in females, then we refer to the process as being oogenesis. But together these two terms fall under the heading gametogenesis, which when it comes to a testo exam, it might be confusing when you have been asked what do we call the formation of gametes through the process of meiosis, then obviously it's gametogenesis, the umbrella name. But if they are being specific, the formation of a sperm cell, then we are looking at the word spermatogenesis. Right, so let's have a closer look at spermatogenesis. Now, we've already done a similar image to this in the section of meiosis, so we already know that we always start with a pre-existing cell, and that pre-existing cell is diploid in nature. So in humans, we will have that diploid number represented as 46 chromosomes. So as we go through the process of meiosis 1, we are going through reduction division, which is then taking the 2n and breaking it down into two cells, each containing the haploid number of chromosomes. So we go from 46 to each cell containing 23 chromosomes. And this happens under the influence of testosterone in the testicles. Okay, so this is only the first half of sperm formation. Then we go through a second round of meiosis 2, as you guys would know by now, where we form 4 fully formed haploid sperm cells. So each one of them will be a viable sperm cell containing all the requirements for life. Now, in a testo exam, obviously there's a lot of information that is linked to the formation of sperm cells, spermatogenesis, and as we've seen in the previous diagram, there are also loads of scientific terms associated with the development of each of those sperm cells. So for instance, primary spermatocyte, secondary spermatocyte, and all of that. But when they ask you in a test, describe the process of spermatogenesis, then the slide that we see on our presentation, this is what we want to write down. So we don't have to go and provide all the sperm cell names. We can just quickly run through it and refer
and
to
Yolandi,
the various...
it's important to note that this is out of the exam guideline for the grade
exactly.
12s.
Yeah. So when your teacher gives you a study examination guideline from the department, this is the exact one that you will see there. So pretty much if you've maybe learned additional information to this process, this is the fundamentals that you would need to know for the examination process. So on the previous slide, we did make reference to obviously all of the sperm cells. If we just quickly go back, to all the names that they go through as they develop, yes, you will still need to know them. So a question could be like a multiple choice question where they ask you which one represents the correct formation of sperm cells from beginning to end, and they could put these names in different orders, for instance. But when it comes down to the actual process, then we're going to talk about how spermatogenesis occurs inside the testicles, and this is under the influence of the male hormone called testosterone. So we are taking a diploid cell, which is found on the wall of the seminiferous tubule. This undergoes the process of meiosis, and eventually we only end up with 4 haploid sperm cells. So it's pretty much as simple as that. We don't have to go into too much detail with it. Right, so when we analyze the structure of a sperm cell, yet again, you can be asked to draw and label your sperm cell. And naturally, you would have to practice this, make sure that all your sections are proportional to each other. You can't have a light bulb sperm cell. Now, I'm speaking here under my own experience, having seen how my students draw the sperm cells, and it's like this, looks a bit like a light bulb at the end. So yes, definitely practice drawing one of these. There are a few diagrams that you might be asked to draw in grade 12, and the sperm cell and the ovum will be one of those diagrams. So when we run through the sections of our sperm cell diagram, we start off with having the head section to it. Then there's a part known as the middle portion or neck. Now, very important to take note of the spelling of this part, section to it. I know some of the textbooks use different labels for this particular section, which I'm not going to mention, but this is the accepted grade 12 label for the middle portion. Right, and then obviously there's a tail section to it, which is this bottom half blue part that we see in the diagram. Okay, so running through the various components, we start off with the acrosome, which is in the head of the sperm cell, and this section contains enzymes. So upon the sperm cell meeting the ovum, the acrosome will open up, the enzymes will come out, and it will digest the pathway through the ovum for the head to be inserted inside it, and that will obviously be the point of fertilization. Then we have the nucleus, which is right below it, and that will be where the haploid genetic information is housed, with all the chromosomes inside it. Right, then we get to the green section here, which represents where the mitochondria is housed. And as you will recall from grade 11 and grade 10, the mitochondria is responsible for producing ATP, energy, and that energy will then be supplied to the tail section, which will allow it to swim. So that will be its way that it will propel forwards through the female reproductive system. Okay, then if we go to the last components of it, the sperm cell also contains cytoplasm, which is severely reduced, and that will allow for sufficient movement or efficient movement. And then it's also streamlined or torpedo shaped to allow for easy movement through the female reproductive system. Right, then we move to the epididymis. Now, this structure here just nestles right next to the testicle component. As soon as the testes produce the sperm cells, the spermatids, they will move into the epididymis. And this is just a temporary storage facility for the sperm where they will mature over the next couple of weeks. And then once it's ready, it will be released during the ejaculation process. So if the sperm cells are not utilized, they can be broken down, and obviously the whole process can start all over again. Okay, so in terms of examination style questions, remember to emphasize that it's temporarily stored and that we don't just say that it's stored there, because ultimately they will be broken down. Okay, from the epididymis, we move up into the next section to it, and that is represented here in the orange, which is our vas deferens. Now, as soon as the ejaculation process occurs, we will find that the sperm will be released out of the epididymis and move through the vas deferens into our urethra and eventually be ejaculated out. So this is also a question that you could experience in the TESOL exam, where they perhaps ask you, what would be the consequence if we had to tie the vas deferens or cut the vas deferens to an individual? Now, yet again, vasectomies is not something that is discussed within grade 12, but they can still ask questions based on it. So obviously, if someone undergoes a vasectomy, vas deferens is cut, and the sperm cells will no longer be able to release out of the male reproductive system. However, there will still be seminal fluid present, but without the actual sperm cells. So just something to keep in mind should it be a topic that is discussed with an examination paper. Okay, so let's look at the glands that assist with the formation of our semen. So we start off with our first one, which is represented here in purple. That's our seminal vesicle. Now, obviously, our sperm cells, they are very metabolically active, and they require nutrients in order to make their way, in order for that mitochondria to actually propel itself forward. So one of the nutrients that we will find will be fructose, and that will provide it with the energy needed in order for ATP to be formed in the sperm cell to swim. Then we have our prostate gland, which is represented here in the blue section, and it will release seminal fluid, which will obviously have a bit of an alkaline buffer to it. It neutralizes the acidic environment of the vagina, so the vagina is actually a very hostile place for sperm cells. And that's just an additional buffer, but it also acts as a lubricant, enhancing the sperm's motility. And then we move to our third accessory gland, and this is the corpus gland, and you can see that it's got attachment place here to the urethra, and its job is to ensure that the urethra's acidic environment is also neutralized, but it also assists with the lubrication, enhancing sperm motility. And then, as we've mentioned, the sperm cells with the seminal fluid together eventually form the semen. Okay. Right, then we have the urethra, which I'm sure that you guys will recall from grade 11. That forms a communal duct for the urine to be removed from the male body, but also an exit point for the sperm cells to move out and semen to move out. So that's very different from the female reproductive system, where there's a definite urethra opening, which is completely separate from the vaginal opening of the reproductive system. Okay, then lastly we have our penis, which is obviously the external male genitalia, and it's richly supplied with blood vessels, which during the process of copulation we would require erection to take place, and that will assist in obviously inserting it into the female's reproductive system and for the sperm to then be ejaculated out.
Right, so let's go over to the female reproductive system, where we're going to look at a lot more detail. So the focus here is long. Okay, so first and foremost, females have 2 ovaries, glandular, meaning that these things release hormones. And we mentioned right at the beginning now, the 2 hormones that are released here are estrogen and progesterone. So these are actually the reproductive structures within the female reproductive system, because not only do the ovaries produce the female hormones, but they also produce ova, which are the female gametes, the sex cell of a female. So let's look at a section through the ovaries to understand this process a little bit better. So here we have an enlargement of an ovary, and we have a fallopian tube and a section of the uterus visible. Now, something that's a bit different between boys and girls is that boys start producing sperm from puberty, and they never stop until the day that they die. So their testosterone levels are fairly high during certain parts of their life and go down towards older age, but they never stop producing sperm. Women, on the other hand, we are born with a certain amount of primary follicles that are capable of producing egg cells. And at the start of puberty, with the first menstruation, we basically start counting down. So every month when you ovulate, you are losing egg cells, and therefore at some point in your life, you run out of them. And seeing as this ovary, in its production of the egg cell, produces the estrogen that makes you a female and that allows your whole menstrual cycle to function, if the ovaries stop producing egg cells, then they also stop producing estrogen. So that's a stage of life which we call menopause, when the ovaries shut down for business and they're no longer producing egg cells and no longer producing the female reproductive hormones. So women aren't fertile for their entire lifespan. You only have what is often referred to as a biological clock, which is ticking down. So if we look at the process here, we say that, right, so the ovaries usually take turns once a month. So you have two of them. 1 month it's the left ovary, the other month the right ovary to produce ova, and sometimes both ovulate, and that's how you actually get non-identical twins. So at the start, we have primary follicles that develop into what is known as Graafian follicles, which is this large follicle containing an immature ovum, and the process through which this Graafian follicle forms and the ovum inside is part of meiosis, and we'll quickly look at that a bit later. So basically, these primary follicles, once a month, one of them will go into this phase, start developing, growing into a secondary and then a tertiary follicle, and eventually have this beautiful Graafian follicle with the ovum inside that is ready to be ovulated. On and about day 14, when LH levels peak, ovulation will happen, where that ovum now is released into the fallopian tube, and you can see that happening there. So only the ovum is released, and what's left of the Graafian follicle will now change into a corpus luteum, which is this yellow body we see in the ovary that's responsible for producing the progesterone, which helps to maintain this endometrium if pregnancy does occur. So here's just a very nice section through an ovary to actually show you the real stuff. So you can see the beautiful primary follicles all sitting here just anxiously waiting to become ova. And then here you can see a secondary follicle, so one of these that has now committed to becoming an ovum or carrying an ovum. It's grown a little bit with the immature ovum on the inside and then these beautiful follicular cells on the outside. And here you can see a mature Graafian follicle, which has this kind of fluid-filled cavity here called an antrum, which allows this to burst, and then it propels this immature ovum into the fallopian tube, and hopefully, if you want to, you can get pregnant if a sperm cell is available. So now, if fertilization does occur, it's the right time of the month, you did have sexual intercourse, then the corpus luteum will remain intact and you will not menstruate. So we don't want to shed this endometrium if you have had that ovum fertilized, because it needs somewhere to implant and to grow. However, if no fertilization takes place, then the ovum, which has now not been fertilized, together with this endometrium will be shed during menstruation. And the reason for this is that the corpus luteum starts disintegrating. You can see here it becomes smaller and it eventually disappears. So the hormones that it's producing to sustain that endometrium is no longer in play. And that then signals the start of a new cycle for the next month. And we'll do the menstrual cycle in depth a bit later. So now, how are ova exactly made? What do I need to know for exams? So like Yolandi mentioned, gametogenesis is the production of gametes through meiosis. Spermatogenesis specifically relates to the production of sperm. Oogenesis now relates to the production of females. And here you can see a micrograph of a beautiful egg cell, and then they enlarge it a little bit to show you that little sperm cell who's got his head through for fertilization. So let's take a closer look at oogenesis. Now, immediately here, you can see some differences between the oogenesis and the spermatogenesis cycle. So where we had a diploid cell in males, the spermatogonium, going through two processes of meiosis, number 1 and number 2, and 4 haploid sperm cells being formed, in females, this is different. The whole point of producing an ovum is to make it as viable as possible. So every time at the end of meiosis, when we go through the telophase phase and you have the cytoplasm dividing, the majority of the cytoplasm will go to one of the daughter cells, producing a polar body on the other side. This again happens at the end of meiosis 2. So this daughter cell, when it divides, will push most of the cytoplasm towards this mature or immature, fully formed ovum that can now sustain a developing embryo, and you have the polar bodies that form. So now one diploid cell produces one haploid cell instead of 4. And that's because the cytoplasm here kind of acts like a yolk, kind of sustaining the embryo. So again, we want to make sure that this is a very healthy, viable egg cell. So as much as cytoplasm as possible goes to one developing ovum. Now, this process is not controlled by estrogen, as testosterone would control it in men. Instead, it's controlled by a hormone FSH, which I mentioned stands for follicle stimulating hormone, meaning it stimulates a follicle to develop. So it's obviously related to the development of this ovum as well. This process occurs in the ovaries the same way that spermatogenesis occurs in the testes. And then that important difference is that one haploid ovum develops instead of 4. So once again, have your exam guideline handy, because a lot of terminology goes into textbooks and they talk about primary oocytes and secondary oocytes, and we're not going to go into that detail because this is the gist of it. This is what you would write if they asked you for 4 or 5 marks to discuss oogenesis. So oogenesis, where does it occur? In the ovaries, and it's influenced and controlled by the hormone FSH. And what does it entail? It entails a diploid cell, that primary follicles, first undergoing mitosis to form more numerous follicles, and then one of these will undergo meiosis, inside which an egg cell will form. So one cell inside the follicle undergoes meiosis to form 4 cells. There you can count them, 1, 2, 3, 4, of which only one survives to produce the mature haploid ovum. So that's a very important distinction between oogenesis and spermatogenesis. Now, just like you had to know the structure of the sperm cell, you also need to know the structure of an egg cell, and you also need to be able to draw it. So on the next slide, what we have is a beautiful ovum, and in exams, always make sure which of these different layers are actually provided. So let's go through them to see where they are. So right on the inside, we have the nucleus of the egg cell, which carries the genetic information of the female. So this will be the female information, and the sperm will carry the male information. And during fertilization, these two combine to form a beautiful human being. Then we have the cytoplasm, which is the fluid part here in which all the organelles will be, and it acts as kind of like a yolk, as I mentioned, to support the development of the embryo. And that's why we want to ensure that the ovum that is ovulated has as much of this as possible. And then around that cell membrane, on the outside of the cytoplasm, we have this jelly layer, which is also sometimes referred to as the zona pellucida. And this jelly layer is the layer through which the acrosome of the sperm cell needs to enter, and provides to some extent protection to the egg cell as well. And in some exams, they also include pictures of the follicular cells. So those are the cells that are left over from the Graafian follicle. Once it has burst open, it pulls some of those follicular cells, called the corona radiata, with it into the fallopian tube. But be careful when you get a picture of an egg cell. They don't always include the follicular cells. So make sure, am I looking at cells, individual like round cells around the sides with nuclei, or are the labels only indicating the nucleus, the cytoplasm, and this jelly layer on the outside? Okay.
Marika, I just quickly want to interrupt you. It's a very good point that you've made there regarding the diagram, because now they don't always indicate it as little tiny follicular cells. Sometimes they put like darker rings around the central nuclei. And I always tell my learners to start labeling from the inside out. And then if you've got an additional ring that you haven't labeled yet, then that will be the follicular cells.
Good advice.
So yeah, definitely, as Marika said, just be careful when you label that one.
Okay. Right, and then continuing on with the structures of the female reproductive system. So we've done the ovaries now, the main reproductive organs producing not only the hormones but the actual gametes. Now we have that ovum being released into this fallopian tube. And again, there are 2 of them. And the fallopian tube has 3 functions. Firstly, this is usually where fertilization happens. So if sperm has been deposited in the vagina at the right time of the month, before ovulation, they would swim up here, hopefully go down the right fallopian tube to meet the egg cell, and then fertilization tends to happen in this area. Then if fertilization happens, this would also be the pathway for the dividing zygote to actually find its way to the uterus. And we refer to a dividing zygote as an embryo. We'll recap these terms again a bit later. And then obviously, being that this is where fertilization happens, the fallopian tubes is also this pathway from which the sperm cells actually enter and meet the egg cell. Then the uterus itself is just a hollow muscular organ, and it can stretch quite a lot to accommodate the developing fetus. In fact, did you know that as a female that has not had a child before, your uterus is about the size of the palm of your hand, and it can stretch massively to accommodate that developing fetus. So hopefully that shocks you a bit. So if you continue here on the slide, you can see a hollow structure, but it has a very important membrane there called the endometrium. And that's what builds up every month and becomes nice and vascular, nice and blood rich, so that it provides a space for the blastocyst to implant and the placenta to form, so that there's an exchange platform for nutrients. As a hollow structure, it not only helps to protect the fetus during gestation, but gives a space for it to grow as well. And this uterus consists of a thick muscle layer that can powerfully contract to push that fetus out through the cervix and the vagina during the birth process. Now, this endometrium itself is going to build up every month during the menstrual cycle. And if you do not get fertilized that month, you can see here in the animation, the unfertilized egg cell, the ovum, as well as this endometrium, this blood-rich layer, is shed through the vagina as blood. And that's what we refer to as menstruation. So that happens in a monthly cycle when fertilization has not happened. Then we have the cervix here at the bottom, which is just the opening between the vagina and the uterus, and it stretches immensely again to accommodate the fetuses moving out during childbirth. When you watch documentaries or series and they talk about a woman being X amount of centimeters dilated, it's referring to how far that cervix has stretched already. And then the vagina is just a muscular tube that receives the penis during sexual intercourse, and it also serves as the birth canal through which the birthing process happens. So through that little hole you have to push a watermelon, girls. It's also the passage for menstrual blood during menstruation, as I've mentioned. So far we've talked about a bunch of different hormones, especially with regards to the female reproductive system. You've heard of FSH, follicle stimulating hormone, so the follicle is growing. You've heard of LH, which stands for luteinizing hormone, because it does two things. It causes this ovulation to happen, and then the remnants of that follicle forms a corpus luteum. Luteinizing hormone, corpus luteum. And then estrogen and progesterone we've mentioned as specifically estrogen helping with secondary sexual characteristics, but these two hormones are very important in maintaining and keeping that endometrium intact, so that if implantation happens, you don't have that miscarriage happening. So let's see how all of these now come together during the menstrual cycle.
Okay, so when it comes to this portion of the work, you need to really have your ducks in a row. So definitely practice this as a type of essay question leading up to your exams. Right, so Marichen has given us an overview in the very first slide as to all the hormones and how they have negative feedback and all of that. And 4 main hormones are of relevance during the menstrual cycle. So the first one being FSH, as we can see in the diagram right here at the top. That is obviously linked to the ovarian cycle. We have the hormone LH, estrogen, and then we have progesterone. So I tell my learners to make a list. Put FSH first, then you put estrogen second, then you put LH down, and then we have progesterone right at the end. So that kind of gives us like a type of guideline as to what is happening when. Okay, so we start off with our follicle stimulating hormone, as Marichen has mentioned. That's taking a Graafian follicle and it literally stimulates development for us. So that's going to be released from our pituitary gland. Okay, so as this now brings about the maturation of the Graafian follicle, we can see that there's also a hormone called estrogen that is being released. Now, this estrogen's job is to go and build that endometrium, making it thick, making it glandular, making it blood-rich, everything that we need in order for implantation to occur. Right, then we move to number 5, and this is now where the Graafian follicle is releasing the hormone estrogen. So as high levels of estrogen is being released, it actually brings about LH from being released from the pituitary gland. So those high levels of estrogen then will cause LH to peak. When LH peaks, that is when we have the Graafian follicle rupturing, so it will border to the wall of the ovary and it will release an ovum from that Graafian follicle. Now the remnants of the structure will pretty much shrivel up, and that will form the structure called the corpus luteum. So the corpus luteum then secretes our next hormone, which is called progesterone. So as estrogen is building our endometrium, progesterone will maintain that endometrium, which is essential, obviously, because if implantation occurs, we need to keep that endometrium intact. So progesterone is vital during the pregnancy component to it. Okay, so progesterone, high levels of progesterone has a negative feedback mechanism, so it will prevent any FSH from being released, and then obviously as a result of that, LH from being released, so we will not have any further graphian follicles being stimulated to form an ovums.
Yolandi, I always like to tell the kids, think of progesterone as the pro-pregnancy hormone. You know, it's rooting for that ovulated egg cell. It's going like, go, 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 no new follicles, no new ovulation, keep that endometrium and let's see what happens.
And it's actually scary because that hormone, that negative feedback system is so vital. Because now imagine if you had to fall pregnant, you have your baby, it's implanted into the endometrium, but your ovaries still kept on making over. And then if you were sexually active during a pregnancy, they will all get fertilized, and you will end up having multiple pregnancies like a couple of months apart from each other.
I literally heard on the radio today about a condition where this happens,
know,
where women can
rare.
Get pregnant after becoming pregnant, but it's like really rare.
Yeah, because then when the first baby comes out, now 9 months later down the gestation period, then perhaps your second child might not be ready yet.
Oh, goodness,
that negative feedback is definitely vital to prevent something from that from happening. Right, so let's get back to our diagram. And we move on. So now we've got our corpus luteum, okay, and it is now shriveled up, and pretty much it will decrease in size. So it will disintegrate eventually over the next couple of days, as we see linked here at the bottom. So as the corpus luteum shrinks, there's no longer a source of progesterone. So we also see a decline in the progesterone levels, which is directly linked to the fact that that corpus luteum is disintegrating. And yeah, pretty much that's it. Okay, so then we have at point 11, we see that due to that drop in progesterone, there's no longer a hormone that is maintaining the wall of the endometrium. So eventually, due to that lack of progesterone, the endometrium will start to break down, and that will eventually separate from the wall, and that is the starting point of your menstrual event. So menstruation will then start. Okay, so as the progesterone levels decrease, nothing suppresses that pituitary gland anymore, and we will find that FSH starts to be released again, and that will be the starting point to a new cycle, a new menstrual cycle. Okay.
So Yolandi, explain to them now, if this is obviously a graph of a woman who is not pregnant.
Yes.
So what are the things we need to look for if the graph was showing a woman who is pregnant?
Okay, so first off, the diagram shows as progesterone is decreasing, right? So that's our first sign that the female has not had intercourse, there's not been any fertilization event. So that's a telltale sign. We can also see from the endometrium that it's starting to break down. So here we see that that endometrium is now the starting point of menstruation. If fertilization had to occur, that would still be like maintained. But also a telltale sign is this corpus luteum, which is busy disintegrating. So we can look at 3 places. The 2 places that you should focus on is whether the corpus luteum remains intact in size or decreasing in size, and what this progesterone curve is doing. So if the progesterone curve drops, no fertilization. If fertilization has occurred, the progesterone levels will just stay high, okay, or will be continue to
and
be
then
secreted.
I know in a previous lesson you also gave the kids a nice tip about if you get these graphs of hormones and they're not labeled.
Yes.
How do you know which hormone is which line?
So you have to be careful with that one, because now, yet again, what they do is it's scenario based. I think that the one that we would definitely can link to is right at the end. If it starts to dip, that's a definite progesterone one, and they can kind of work backwards. Usually with LH, that coincides with day 14, where it will be at its peak. So you can look for LH as well. And then, as I said, I normally tell my learners to number the hormones. So we start with FSH. So usually that's the one that peaks first. And then as a result of FSH being released, estrogen starts to peak as a result of estrogen being released. That stimulates the release of luteinizing hormone. So you can kind of work your way forwards or backwards, whichever way around. But look at the scenario that they sketch for you.
I also want to give the tip that with regards to estrogen's graph, it's the only graph that peaks twice.
Yes.
So you see the peak in the first half of the cycle and in the second.
Which coincides with the increase in FSH.
There we go.
Yeah.
And the progesterone peaks towards the latter part, so the second part of the cycle. So yeah, make sure you go through many examples of questions, but this is definitely a section that they like to ask questions on.
They can either ask it as an essay question or scenario-based question, asking whether the female has fallen pregnant or not, or they can give you a diagram and you have to answer questions based on the diagram or the graph, and then obviously write a little mini essay on that topic as well.
Yep.
Oh,
Okay.
right.
So let's move on to fertilization, implantation, and gestation. So first off, let's see the ovary here. We are now basically looking at what's going to happen after fertilization does occur successfully. So here we have the ovary once again, and we're going to look at the different stages in this diagram. We're going to break it down. So firstly, ovulation, right? Ovulation is when high levels of LH, as Yolandi has mentioned now, triggers the rupture of that Graafian follicle. That fluid-filled cavity called the antrum will rupture, pushing that ovum out, and you can actually see it beautifully illustrated here. So there's the little ovum and some of the follicular cells in black coming out with it that forms the corona radiata. So ovulation is the release of an ovum from the ovaries into the fallopian tube. Now what happens is fertilization. So if you've had sexual intercourse at the right time of the month and the sperm are there meeting the egg cell, this haploid sperm cell containing 23 chromosomes will fuse with a haploid ovum carrying 23 chromosomes, creating what is known as a diploid zygote. So zygote is the single cell with 46 chromosomes just after fertilization has taken place. That zygote will then start dividing through mitosis. So now we are done with meiosis. Meiosis produced gametes for us. It halved the chromosome number, produced variation and all the things that it's good for. But now after fertilization, we have to grow. And growth is done through mitosis. So they like to ask the steps now, okay, and being able to recognize the different stages. So here we have a zygote with a single nucleus with 46 chromosomes, and it starts dividing through mitosis into a 2-cell embryo. So an embryo refers to the dividing zygote, and it has different stages that are recognizable. So those 2 cells will then pop into 4, into 8, and then bloop, 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 bloop. It eventually forms this beautiful dense ball of cells called a morula. I have a video that the guy says it like this. Love it. Sounds like Dracula. Did you know morula means raspberry in Latin? So it's because of all the little cells that make it look like a raspberry. So that's a morula, a dense ball of cells. And that then continues to divide to form a hollow ball of cells called a blastocyst. And this will be about 5 to 7 days after fertilization. This is happening as it's moving down the fallopian tube. And the blastocyst is the one that ultimately implants. So you can think now if day 14 is ovulation, about 5 to 7 days afterwards, and then 2 weeks later is when you start missing your period, right? And you think, okay, maybe something is wrong. I might be pregnant. Now, if we look at the blastocyst here, you have to describe this as a ball of cells, a hollow ball of cells, and the morula as a dense ball of cells. So make sure you go to your exam guideline to make sure of that division process, because I say they love to ask this for 5 or so marks, the development of the embryo from fertilization until implantation.
And it sounds very daunting, and
Yeah.
it's actually just like
It's
a diagram
so easy.
order. Yeah.
So here's a little animation to actually show you these steps. So there's the zygote, 2 cells, 4 cells, 8 cells, morula, and then the blastocyst starting to form. And the blastocyst is a thing that implants, and we'll talk a bit about implantation at a later stage. Now on the next slide, I'm going to show you a video of this actually happening under a microscope, and I want you to look at a few important things. So the first thing you're going to see is the zygote. And you're going to see it as a zygote because you're going to see two nuclei that suddenly appear and then disappear. And then the cell bloop goes into 2 and then into 4, and then it starts dividing into this dense ball of cells. And obviously this has been sped up over a course of days now, so it's not happening at that speed as we're looking at it, but it just shows you all the awesome stages of development. So are you ready? Let's go to the slide. Right, so there you have the two nuclei and boom, they disappear. 2-cell embryo, 4-cell embryo, and then it divides, bloom, 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 bloom. And then it forms a dense ball of cells, your morula, and then that's the start of one of the stages of your blastocyst. So that's actually where they get these images from. It really looks like that. Okay, and that's called hatching, which is part of the implantation process. So now Yolandi takes us further for implantation.
Right. So the diagram that we see here in front of us, we can now look at how that little structure, the blastocyst, has embedded itself into the wall of the endometrium. Now, in one of the previous pictures, they actually showed the blastocyst with having these little spike-like structures that stick out. They are called chorionic villi, and we also like to refer to them as being finger-like projections, which will eventually form the outer membranes of our little embryo. So the cells that we see here, that will be our starting point to our embryo developing, and eventually it forms into a fetus, and eventually upon the birthing process, we would refer to it as a baby. So it goes from blastocyst, embryo, fetus, baby, as we continue through the developmental stages of gestation. Right, so gestation is obviously what we call the period in which the fetus develops inside of the uterus. And this quite often gets misconstrued for the process of pregnancy, which includes all of the hormonal changes that takes place in the body, as well as what happens to the fetus. So gestation is purely the period of development of the fetus inside of the female. So we know that period to be about 9 months in whole for a little human to form. Right, so in terms of what surrounds the developing fetus, so here we can see the little gray area, that's the amniotic fluid, and surrounding the amniotic fluid we've got the amniotic sac. So I like the fact that they've actually called the fluid something that relates to the membrane that immediately surrounds it. So that's the amniotic fluid and the amniotic sac. And pretty much the functions of it is firstly for the fetus to practice movement in. So we have all seen how sometimes when the baby kicks, you can actually see the woman's abdominal area like distending where the foot is pressing against it. So it allows the baby that freedom of movement. So it's not constricted with inside the uterus cavity. Then it protects the fetus against temperature fluctuations. So very important to just relate here that the fluid doesn't have its intrinsic ability to make the fluid warm or cold or anything, but it does allow for like a buffering system. So if the mother is cold, then it will take a while for that cold to actually seep down to the fetus. So that fluid is a temporary like buffer situation for the baby, just to protect it against the cold. So no intrinsic ability to heat up or cool down or anything like that. It is still linked to the mother's external temperature as well. Okay, then we move to our next bullet, which is to protect the fetus against dehydration. And lastly, it acts as a shock absorber. And quite often my learners ask me, but how's this a relevant point? So I tell them, well, imagine if someone had to push you and you didn't have anything surrounding it, you will kind of like just fall forward, right? Now, if you were in a swimming pool, let's say you're a polo player and someone had to push you, would you just like fly to the edge of the swimming pool? And the answer is no. You would maybe like float to the next player next to you. And that's exactly what happens inside of the woman's amniotic sac as well for the little fetus. So if the mother had to trip forward, you don't see the baby hitting the wall, okay? It will move slightly and maybe like whoop, whoop, slightly bump the side of it. So it's a little bit of a shock absorber protective fluid that it is obviously surrounding the fetus. Okay, so then we move on. Then we have the chorionic villi, which we've seen in the blastocyst, little projectile villi, finger-like villi, that will eventually embed itself into the wall. And that, along with the endometrium, will form the structure which is called the placenta. And as we can see here in the placenta, we've got the mother's circulatory system, which runs in a very close association with the circulatory system of the placenta and ultimately that of the fetus. So there's no direct link between the mother's blood and that of the fetus. So obviously the aim there is to act as a type of filtration system.
Yeah.
Okay, so when we look at the placenta, I like to make the association. It's like a charging cable that you put into the wall, and out of that plug, all the electricity would run to your device. Okay, so the same thing happens for the fetus. The placenta is the plug, and the umbilical cord is like the cable wire that runs to your computer, but now on the other side, instead of a cell phone or a laptop, you're going to have the fetus. So that is what the baby would need in order to obtain nutrients and oxygen from the mother. And at the same time, that fetus is metabolically active. So it's still producing carbon dioxide, it's still producing waste products, and all of that needs to be returned to the mother. And that happens through the placenta. So at that barrier, all the gases will move across, all the waste will move across, and the nutrients will move across. And pretty much the mother ends up excreting for two, respiring for two, eating for two,
So
urinating
you are a
for
little
two.
parasite.
That's exactly
That's
where
what
I
it
was
boils
getting
down
to.
to.
So yes, you are a little parasite sucking your mother of all the nutrients.
And some of you don't stop until you're like 30. Eh?
So when we look at the functions of the placenta quickly, and we do a quick run through them, one of the first things is then obviously the diffusion of nutrients and oxygen from the mother to the fetus. Then we also have the diffusion of carbon dioxide thanks to fetus undergoing cellular respiration and waste products from fetus to mother. So that's where the mother breathes for two and excretes for two. Okay. Then it also serves as a microfilter, preventing any pathogenic substance from getting into the bloodstream of the fetus. But unfortunately, that microfilter does not have a 100% success rate. So if the mother is abusing drugs or alcohol, that can still filter through into the bloodstream of the fetus and affect its development. Okay, and then lastly, and most important of all, the placenta takes over the role of the corpus luteum about 3 months into it and starts to secrete progesterone. And that is essential, as we've established, to maintain the endometrial wall and ultimately to maintain the pregnancy. So that is a
interesting
vital
fact about that, Yolandi, is that handover between the corpus luteum and the placenta, the 3-month stage, which is usually the end of your first trimester. And that's when many miscarriages actually happen, because it's like a relay, you know, you let the stick drop. So that's why they tend to say, you know, don't tell people you're pregnant until about 3 months have gone by, and you know everything is fine.
That the placenta has been fully formed and it is actually maintaining that endometrium for them. Yeah, very good point there. Okay, and then ultimately when we stretch out our structure here we can see what the placenta looks like with our fetal blood network supply running from it, going through the umbilical cord to the belly button of the baby. And this consists of obviously 2 blood vessels, the first one being the umbilical vein. Now, you would recall from your circulatory system chapters in previous years that veins usually carry deoxygenated blood, but because we have a blood network system, a blood vessel going into the fetus's heart, okay, we have a vein going into the heart, then it has to have oxygen going to the baby, right? So it kind of swaps over functions here when we talk about veins usually carrying deoxygenated. Here, the umbilical vein carries oxygenated blood, but it's also very rich in nutrients. So as it heads into the offspring, into the fetus, it will be supplied of everything that it needs. But just as we require oxygen and nutrients, we also have waste products which needs to go from the fetus to the mother. And in that case, we've got our umbilical arteries. So there we now have deoxygenated blood, and all of the waste products that the cells have produced in the fetus, and that will then
So
diffuse
the
across to
emphasis
mother.
then with vein and artery is not what blood they carry, but rather what direction.
Exactly.
Okay.
Right.
So that's basically it. A lot for 55 minutes. We've given you an overview, so it is a lot of information, we know that, but hopefully you have kind of the basic, like, big picture idea, and we filled some gaps that you might have been wondering about.
And if you have a look at the PowerPoint presentation that we've used, there are some past paper questions, and also your resource booklets will also have a few past paper questions in it, and definitely worth spending some time on that and working through them as well.
So we'll see you again for the nervous system.